Can everybody hear me?
Yeah, we can.
Okay. Well, we're going to start our final lecture of the semester, formal lecture anyway. If anyone has any questions about any of the material, you can ask it at the end of today's lecture, which I've significantly shortened. Or you can send me an email and I will respond. Just to remind you, the third exam is coming up, and we're going to talk about it a little bit after the formal presentation. And, you know, be glad to address any questions again after the formal presentation is finished. But the exam, third exam, I expect you all to turn it in by 1 o'clock Friday. I said I was going to send it out on Thursday, but I may actually send it out on Wednesday afternoon to give you all a little bit more time. Again, we're going to adhere to the honor system, which I think worked fairly well last time. And we'll talk about the exam questions and how difficult I think they are relative to the first two exams a little bit later on. Today, we're going to talk about what I think is one of the most amazing events that occurs in biology, and that's metamorphosis. We're also going to talk a little bit about the plasticity of the genome, which is to say that the genome can respond to different environmental or other factors and result in a completely different morphology. So the genome contains within it information regarding basically several different forms, if you will. This can be illustrated with butterflies. The European map butterfly has 2 seasonal phenotypes. The pattern is determined by a combination of daylight as well as temperature. When the species was originally captured in Europe, they actually thought it was 2 different species because they're morphologically very distinct. It wasn't until they started growing them in the laboratory and could manipulate both daylight exposure as well as temperature, they realized, in fact, they are the same species. And I'll show you a picture in just a minute here. Yep, as I said, in the lab, you can actually mimic the conditions and generate either of the 2 morphs. Here you can see the pigmentation and everything is vastly different. The body shape and everything is very similar, but morphologically distinct. Summer, longer days, higher temperature. Spring, shorter days, lower temperature. It's not just the adult stages either. You can also see it in larval stages. This is a moth. You can see that those that are growing in springtime very much mimic oak catkins, which are prevalent in spring. Leaves really haven't yet come out. The summer one looks like oak twigs. You can see the morphology is very distinct in this larval structure. And you can convert one into the other based on diet. The default state, if you will, is this spring form. But if you feed the spring form oak leaves, which is what the summer one is consuming, it will actually convert into the summer morph. Plasticity of the genome. Mexican axolotl, if you haven't seen them, they're fascinating creatures. They're found mostly around Mexico City. In the wild, they're somewhat endangered, but they've been used and bred a lot in laboratory settings, as well as you can buy them in pet stores. Pretty easy to breed. In salamanders, you see what's referred to as neoteny, which basically just means slow development. We show that as well. We don't mature as rapidly as some of our predecessors. In the case of axolotl, there's a thyroid stimulating hormone that is not produced. The pituitary does not produce this thyroid stimulating hormone. So no T4, which we'll talk about in a couple of minutes, is produced. This is one of the triggers for inducing metamorphosis in amphibians. It does, however, make the receptors for this hormone, T4. So there's a couple of different ways you can force the Mexican axolotl to undergo metamorphosis. In the wild, it would never do so, but through diet or injections, thyroid hormones, even iodine, and you'll see how iodine fits in just a couple of minutes, can result in an organism that you would never normally see in the wild. On the left is the normal axolotl, the larval form. They reproduce as larvae. On the right is one that has been fed or injected thyroid hormones or iodine. You can see vastly different morphology. Again, genomic plasticity, the information is there to undergo metamorphosis, but through evolution, they stop producing, pituitary stopped producing the hormone that would stimulate the thyroid. There's a cost associated with growing up, at least for axolotl. One of the reasons they've been so heavily studied is that they have remarkable regenerative abilities. You can lop off a limb and it will regenerate another limb. Occasionally, it will actually repair the old limb if it's not completely removed and grow an additional limb. So they'll actually end up with 5 limbs. That's in the larval stage. If you force them to undergo metamorphosis, this ability is substantially reduced. And they can regenerate part of their brain as well. I mean, it's remarkable, everything that they can regenerate. And that's been the reason why they've been so popular in laboratory studies. It also has a cost in terms of lifespan. The typical lifespan, if they're allowed to remain in the larval stage, is 15 to 20 years. If you force them to undergo metamorphosis, it goes down to about 5 years. So you can see that there are times when it actually pays not to grow up. Peter Pan. Metamorphosis or development can be divided into 2 fundamental groups. You have direct developers, just what we are. Young are basically smaller versions of the adult. And over time, we just continue to grow. There's no dramatic metamorphosis that occurs, although we do undergo a metamorphosis. We call it puberty. And then you have indirect developers. These are the vast majority of animals, because the vast majority of animals are actually insects, where the larval stage is significantly different than the adult. And we're going to focus mostly on amphibians and insects that undergo a complete metamorphosis when we talk about these things. Typically, the larval form is very specialized, either for growth, feeding, or for dispersal, depending upon the particular species. The adults are primarily there for reproduction. And in fact, some of them, and we'll talk about a couple, can't even feed themselves. So once they undergo metamorphosis, their lifespan is not very long. As I said, most animals, again, we're talking insects, the larval stage is strikingly different from the adult. It's true of, it's true of amphibians as well, as you can see from the axolotl. By the way, as I said, I think you can buy the axolotls at pet stores. And as long as you don't feed it thyroid, it'll remain aquatic and it will live for as long as typical dogs do, longer than many. As I said, the larval forms are often very specialized, typically either growth or dispersal. Cecropia, moths. It's the largest moth in North America, 6-inch wingspan. The good news, after months of gorging themselves as a juvenile in the larval stage, caterpillar, you're ready to mature into an adult and now you can mate. The bad news, you're not going to have a second date next month. Their lifespan, once they undergo metamorphosis, is about 2 weeks. They're incapable of feeding themselves. And that's true of a lot of insects, where the adult stage, the only function is to mate and then die. Now, this process, as we've sort of alluded to already, is regulated by the production of hormones. Oftentimes, it's the same hormones that were used during embryogenesis are once again expressed to undergo or initiate metamorphosis. Oftentimes, significant morphological, physiological, and behavioral changes occur. I know when I went through puberty, I had a very tolerant cousin. My hormones, testosterone primarily, I'm sure were fluctuating wildly, as was my voice. And so occasionally I would use the same effort that I had used before undergoing metamorphosis, puberty, and I would end up with door handles in my hand. One cousin, I broke the handle to a rototiller, I broke the handle off his van, and I broke the handle off of his refrigerator because I hadn't yet figured out how to apply the proper amount of force. So even in humans, can be significant changes. Amphibians literally means double life. Amphi meaning double, bios meaning life. They have a double life. Typically one is aquatic and the other is terrestrial. Aquatic to terrestrial lifestyle. And this is, I think, one of the more fascinating things of biology, particularly developmental biology, is this transformation from aquatic to terrestrial lifestyle. You see it occurs in a lot of insects as well. Mosquitoes, for example, the larval stage is in water, which is why they say if you want to try to eliminate mosquitoes, don't have old tires lying around, they're going to collect water, or anything else that's going to have stagnant water, because it's a great place for the mosquito larvae to grow up. And as we all know, the adults can fly. In the case of salamanders, as well as frogs, you see reabsorption of the tail, destruction of external gills, and skin modifications. Frogs, it's even more dramatic than in salamanders. And we're going to go into a little bit of detail regarding what all goes on in frogs, with frogs. I don't expect you to know all these things. I just want to provide you with this. You can see in the case of anurans, which are frogs and tadpoles, locomotion is altered, respiration is altered, the circulatory system becomes altered, nutrition, what they feed upon, becomes altered, the nervous system, excretory, and the epidermis changes. Lots of changes occur. Virtually every system gets modified in going from a tadpole to a frog or a toad. It's initiated by thyroid hormones like trioxin, which we'll refer to as T4, or threonine, T3. The larval organs, again, some of them will grow. They'd stop growing during the larval stage. Now they resume growing. You'll see death, apoptosis, remodeling, or respecification, going from one function to a different function. T3 causes the limbs to emerge. A nictitating membrane, which is a transparent eyelid that develops so that when they go from a aqueous environment to a dry environment, their eyeballs won't dry out. Proliferation and maturation of neurons as well. So T3 induces a lot of things. The eyes move from the lateral to the front. They go from the eyes of prey, which is what most tadpoles are, where you want to have as broad a vision, as big a field of vision as is possible to see any approaching predators, such as a bass or a snake or something, to those of a hunter, where depth perception is critical. And frogs eat predominantly insects. As a predator, they need binocular vision. They need depth perception. The tadpole, the right eye innervates the left side of the brain and vice versa. They don't have same side projections. They don't have ipsilateral projections, so they can't have a depth field. You got a field of depth. During metamorphosis, however, the ipsilateral projections emerge. Both eyes input images to both sides of the brain. This allows for depth perception. In Xenopus, at least, this event involves remodeling, but does not involve, remodeling, but the generation of new neurons. Mammals were born with ipsilateral vision, but not true of fish or birds, or many amphibians. Here you can see the difference between the eye location, between the tadpole on the side of the head, giving almost 360 degrees vision, to that of a frog, where it generates depth perception. So you want to be able to run away from predators, and then you want to become a successful predator. And you do that through ipsilateral neuronal connections. T3, the hormone, causes cell death in some structures. The tail, gills, for example. Early death is typically due to apoptosis. Some of the later death occurs by something other than apoptosis. The tadpole red blood cells are also all going to die, and they're going to be replaced with adult ones, whose hemoglobin binds oxygen more slowly, but releases it faster. In humans, we have several different types of red blood cells. Some of the earliest ones we have are actually very good at taking oxygen from mom's blood. So we actually steal oxygen, and then once we become an adult, our the binding capacity of the red blood cells changes. These responses are tissue specific. It's not location on the body that determines the fate of the cells. You could take a tissue that is normally absorbed, transplant it to a part of the tadpole that doesn't die, such as the head, and nevertheless, the tail will regress and go away. So once it's exposed to certain levels of the hormone, it knows its duty is to largely complete apoptosis and get rid of its tail. You don't want to be jumping around on land with a tadpole's tail, so it gets reabsorbed. Likewise, you can take a tissue that is not destined to be destroyed, like the eye, and transplant it to the tail, a tissue that typically is reabsorbed. And you see that although the surrounding tail has been destroyed, the eye nevertheless remains. So it's a tissue-specific response, or based on instructions that were established some time ago. Yeah, transplanted tail regresses, and the eye remains. Remodeling the intestines. Most tadpoles are actually vegetarians, vegans. In order to be successful at digesting, you need lots of intestines, much longer coils than what is needed by a carnivore. And so the intestines become much shorter during metamorphosis. Old extracellular matrix becomes degraded, several different morphogenic proteins expressed, and various genes get turned on. I don't care that you know the names of these proteins, because we're not going to talk about these signaling proteins very much. No, I'm not going to talk about them at all. It's thought that these different proteins trigger apoptosis as well as remodeling. Paracrine factors, you may remember these are signaling compounds that interact with receptors, but they do so over a relatively short distance, up to around 20 cells, something like that. They start to undergo metamorphosis, being triggered by secondary events after T4 and T3 have arrived. They are hormones produced in the thyroid and then diffused throughout the body, as opposed to paracrines, which are relatively local. The nervous system, the optic nerves, tadpole jaw converts into an adult jaw. Innervation of newly formed tongue. Tadpoles do not have a tongue. And I'm sure we've all seen nature videos or YouTubes of some amazing frog tongues and how skilled they are at plucking insects out of the air. They don't even have a tongue until they start to undergo metamorphosis. The neurons for the tongue are present, but they're dormant. They don't form their first synapses. They don't connect with other neurons until metamorphosis is initiated. The lateral line system degenerates. This is a system that is employed by aquatic organisms to detect water movement as well as pressure. That is how far down they are or how high up they are in the water. The lower you go, the greater the pressure. So it's water movement as well as pressure. Those aren't needed on land, so the lateral line system degenerates. Just tremendous restructuring that's going on. The skeleton, tadpole skull is mostly neural crest-derived cartilage cells, whereas in the adult, it's neural crest-derived bone. The lower jaw, cartilage nearly doubles in length, and the dermal bone forms around it during metamorphosis. Skeleton, some cell death, some proliferation, some remodeling. Whether you realize it or not, our bones are constantly degrading and regenerating themselves, osteoclasts and osteoblasts. And when those get out of sync, that's when certain forms of arthritis can set in. Biochemical changes. The tadpole liver generates ammonia for excretion of waste products. It's very typical of freshwater fish. But as an adult, they switch over to urea because urea requires far less water. But if you're living in water, fresh water, then you can afford to use ammonia instead of urea. T3 induces transcription factors, stimulating the urea cycle while repressing those for ammonia, either directly or indirectly. As I said, hormonal control of metamorphosis, just as in us, was first demonstrated in 1912 when Germans fed horse thyroids to tadpoles and forced them to undergo premature metamorphosis. Why they would want to do that, I don't know. But that was the first evidence that something in the thyroid was inducing metamorphosis. Also talks about how evolutionarily conserved some of these hormones are. If you do sort of a reverse experiment, if you cut out a tadpole's thyroid, it just continues to grow as a tadpole. It never undergoes metamorphosis. So you end up with a giant tadpole. The responses are regulated by the amounts of the hormone, both hormones. Limbs start to be made at low concentrations of the hormone. Tail loss does not occur until the concentrations are high. So you start off low and then you go high, which makes sense. You need to have functional legs before you get rid of your tail. Otherwise, you become very easy food for bass and whatnot. I'm going to talk about the threshold model, which is somewhat simplified. And the idea is that events are triggered by different levels of hormones. As I said, it's simplistic. It's a little more complex than that, but it gives you an idea of what's going on. You first have the release of T4 by the thyroid. Then you have conversion of T4 to T3 in target tissues. T3 is the more active form. We're going to talk about that a little bit. In some tissues, you actually have degradation of T3. So you don't have a response to it. T3 binds to nuclear thyroid hormone receptors with a higher affinity than does T4, TRs, these receptors. And the TRs, these receptors now become transcriptional activators. I can't seem to pull it down. Oh well. The last line says TRs become transcriptional activators. The concentration of T3 is regulated by T4 initially in blood and by two enzymes, intracellular enzymes, which remove iodine. This is where iodine comes in. Remember I said if you inject axolotl with iodine, you can artificially induce metamorphosis because iodine plays a very big role in this process. Type 2 deiodinase removes one iodine from T4, converting it into T3. If you don't want to respond to the hormones, there's another deiodinase, type 3, which removes an iodine from T3, causing it to be inactivated, and ultimately it's going to be broken down into tyrosine. So it's one way to insulate or prevent other tissues from undergoing a response to the presence of these hormones. You also have two types of receptors, thyroid hormone receptors, alpha and beta. Alpha is present in all tissues at the beginning before the thyroid gland even develops. The other receptor, TR beta, is the product of a gene directly activated by the thyroid hormones. So you're ramping up the receptors to pull in more of the thyroid hormones. Of course, levels of TR beta, initially low, but it increases as metamorphosis proceeds. These receptors form dimers with retinoid receptor, RXR. This association, TR-RXR, is already associated with genes before T3 is actually generated. And this is a very common thing in the process of development. You often have transcription factors associated with enhancers that initially act as repressors until another cofactor comes along and turns them from repressors to enhancers. This is what's going on here. So we have them pre-positioned, associated with genes. It by itself, TRRXR, recruits histone deacetylase, which means the chromatin is going to be more highly compacted. And as I said, it acts as a repressor of gene expression. The binding of T3 to this other association, so T3 TRRXR, converts the complex from a repressor to an activator. And one of the things it does is to recruit histone acetyltransferases. So now you're opening the chromatin back up to allow for transcription. And we see this quite a few times during the process of development. You have prepositioned transcription factors awaiting a third factor to come along and signal the transition from repressor to an enhancer. In terms of metamorphosis, 3 different types. You have direct development. After the early pro-nymph stage, the juvenile looks just like the adults. I'll show you some examples of these in insects. Hemimetabolous, which is a gradual change. With each stage, the nymph resembles the adult much closer. So it starts off different, and then through each molt, in the case of insects, it gets closer and closer to the final adult version. And then you have holo, meaning complete, metabolous. This is complete metamorphosis. The larval and the adult form differ greatly. And you can say that's true of the amphibians, and it's true of Drosophila melanogaster, lepidopterans, the butterflies and the moths, several other, the hymenopterans also, bees and wasps and whatnot. Here's an illustration. On the left, you can see that the adult is essentially a larger version of the pro-nymph. That is the thing that's come out of the egg. Here we have a cockroach. It starts off somewhat resembling the adult, but no wings, for example, and those evolve and develop eventually. So each larval stage, each molt, results in a creature that morphologically looks more similar to the adult. And then we have the things that we're probably most familiar with in terms of metamorphosis. In this case, it's a moth. You can see the initial larval stage and all subsequent larval stage are morphologically very distinct from the adult. Typically, these that undergo complete metamorphosis do so in some kind of a protective case, if you will. A cocoon in the case of moths, and a pupal case in the case of dipterans. Flies, mosquitoes, things like that. Okay, that's just sort of hitting the highlights of metamorphosis. What I think is most fascinating is that metamorphosis for insects in many ways is a lot easier than it is for amphibians. Amphibians need to maintain everything while trying to avoid, they have to feed themselves, they have to avoid predators, all while undergoing this massive transformation that we've talked about. So they can't afford to simply hunker down and undergo metamorphosis the way insects do. Insects, arguably the metamorphosis, the transformation, is more profound. Almost all larval structures are totally destroyed, not entirely. Some of the CNS, some of the neural tissue persist and things like that. But for the most part, almost everything is totally destroyed. And the adult arises from a different subset of cells. The amphibian, on the other hand, has to gradually make modifications. As I said, everything has to be timed properly, which is why the threshold model comes into play. You don't want to start absorbing your tail too soon. Otherwise, if you can't swim and you can't walk on land, you're dead. So that's the material. we're going to end here. It's a little shorter than I thought it was going to be. You've got other additional information if you have any curiosity, if you want to see about how Drosophila melanogaster goes from a larvae to a pupal case to an adult. There's information on that. Basically, larval growth in insects isn't so much through cell division, rather each individual cell actually gets larger. At metamorphosis, however, you have cells that have been mitotically active all along, but slowly. You have a group of cells called the imaginal disc. It's after the German word imago for adult. These imaginal discs are sequestered very early during development. They continue to divide during the larval stages, but otherwise make no contributions to the developmental state of the larvae until metamorphosis. And in metamorphosis, they stop dividing and they start to assume their adult structures. All of the external features that you see in melanogaster, with the exception of the abdomen, are derived from these imaginal discs. So just a review of the chapters we're talking about. We have sex determination in Drosophila melanogaster, as well as CRISPR-Cas9. We went into a little more depth, and a little bit more on RNAi. That was the first set of PDFs that we went through. We talked about forensic DNA. We also talked about some of the limitations. If you haven't listened to the YouTube that I sent you all, that was in the notes, John Stewart, please listen to it. It's hilarious and very informative. We also talked about horizontal gene transfer, something that we're now recognizing with genomics and whatnot. It's a lot more common than we ever originally thought of. And horizontal gene transfer can also involve things like matings between Neanderthal and Homo sapiens, or Homo sapiens and Denisovians. You can also have horizontal gene transfer through transposable elements. So there's several different ways that you can introduce genes from a different species into another genome. We talked about HIV, a disease that's still way too prevalent, especially in Africa, but other places as well. It tends to be a disease of poverty, associated with poverty and ignorance. The one very common myth in South Africa, at least, was that the way to cure HIV, which, by the way, there is no cure, there are treatments, but no cures. One way to cure yourself of HIV was to have sex with a virgin. And it resulted in just a tremendous outbreak of young females being raped by males trying to cure themselves of HIV. Of course, it doesn't work. It just doesn't work. Just like we currently have no treatment. For HIV, we do have a treatment. We don't have any treatments right now for the coronavirus, COVID-19, that is circulating around. There's lots of different drugs and things like that being tried and are in various clinical trials and whatnot. But thus far, nothing has actually been proven to be effective against it. As I said, with HIV, though, it also, about 50% of the people, the treatments work. 50% of them, they stop working after about 3 months. We talked about cancer. We talked about how cancer can be caused by one of two fundamental ways. There's a couple of other, but primarily, most cancers are either due to loss of a tumor suppressor, such as retinoblastoma, genes that are involved in maintaining checks on the cell cycle so that they don't progress through the cell cycle. As long as you have one good copy, that's sufficient to prevent cancer from forming. To get cancer, you need what's called the two-hit hypothesis, which is where both copies have to be destroyed. So tumor suppressors are recessive mutations. When they become homozygous mutant, then you can have cancer develop. We also talked about oncogenes, which is a dominant cause of cancer. And we talked about the Philadelphia chromosome and the translocations and some treatments that have been designed based upon understanding the three-dimensional structure of the cancer protein. And we talked about ways to identify oncogenes. And then finally today we talked about metamorphosis, which if we had more time in the semester, we'd go into greater detail. As you can tell by the rest of the PDF that I sent you, there's a lot more information, but I just wanted to touch upon it because I think it's one of the more fascinating things in biology. It's just remarkable, particularly amphibians, the way they still survive in a very active, very dangerous environment for them, and yet are undergoing just dramatic changes in virtually every system they have. But it has to be gradual changes. Okay. Any questions thus far on either metamorphosis or any of the material we covered? Anything?
Is the exam not going to be over the systems biology lecture then?
Oh, sorry, I forgot about that. No, you're right. Systems biology is also part of it. Sorry, I forgot about it. Which is a very important topic right now with all the efforts to try to develop treatments for the coronavirus as well as vaccines and whatnot. Good point. This is sort of the wave of the future, systems biology, which is where biology is sort of moving into big science, if you will. Biology used to be individual labs were sort of standalone with their own results, their own experiments, their own hypotheses. And now we're having to integrate everything if we're going to understand how system biology works, how the whole is actually greater than the sum of individual parts. It's the area where reductionist biology, which is what I've been most of my life, what most people have been up until recently. Reductionist biology was great in identifying the partners, the players, but it really could not inform us about how they integrate together and end up modifying each other so that the total ends up being different than what you would anticipate simply by looking at destruction of individual components. Good point, thank you. Anything else? The exam is the format that we've used in the past. There are 6 10-point questions. 1 or 2 of them are fairly easy, I think. And a couple of the other ones are more challenging. I think overall, it's probably going to be a little easier than the second exam, but it will be more difficult, more challenging than the first exam, in part because the material is more complex, but also you all are going to have more time in which to answer it. I sort of view the 10-point questions as almost a take-home exam where you're not constrained by time. The true, false, and why, however, you should think of as you only have a finite period of time to try to complete them. And there's 10 true, false, and why. You get a point for getting it correct, true or false. But 3 points, the majority of it, 75%, it comes from being able to defend why you think the statement is either true or why you think it is false. Usually it's easier to explain why something is false than it is true. And there's a mix of both on the true, false, and why. Any questions?
We talked about 5 or 6 different specific cases. Do we need to know all of those?
By cases, are you talking about forensic?
Yeah.
Okay. No, not really. You should take away the general information, how the STR works, how things can go wrong, the complexities of doing DNA analysis, especially with mixed samples, why you get allelic dropout or why you might get other defects or other problems in STR analysis, why it's so critical to have the proper concentration of human DNA. It really only works within a certain range, and that's true of all techniques that you're going to use. It's something that's probably not emphasized enough in undergraduate education, but every machine you work with, every technique you're using, typically is only valid within a certain range, certain concentration of reagents, certain intensity of material or energy. You go outside that and it's no longer linear, you no longer get a linear response. And so STR analysis, that's true also, which is why determining the amount of that is important. Now, I gave you in the forensic DNA, I want you to know that it's not just limited to humans, that you can derive information regarding, if you think about it, what forensic DNA really does is it links somebody to a particular space. It says that that person was present at this space prior to crime scene investigators collecting the evidence. But it doesn't have to just be human DNA. It can be, as we talked about, Snowball, the cat, how the presence of this perpetrator's cat hairs on his victim indicated that indeed he had been there, because otherwise the murdered individual, female, would not have had the cat hairs, the specific cat hairs on her person. Also, trees, pods and whatnot have been used to indicate that somebody actually had been to the scene of crime. So what DNA does is it links somebody spatially. It doesn't link them temporally. It doesn't say anything about the timing, but it does link them to the particular area. I also, the takeaway from the Thomas Jefferson thing is don't overinterpret your data. The forensic DNA, STR analysis, is often called the gold standard in the field of forensic science with some justification, but it does also have its limitations. And that's what the TJ thing was about. It's also interesting, but again, I went to UVA. If you go down there, you hear UVA referred to as Mr. Jefferson's University. You also hear him referred to as TJ. He's held in a lot of very high esteem. But like all people of his time, like all people of our time, we have our flaws. So it was to illustrate that, but it was also to illustrate limitations. That you need to bear in mind, again, limitations of your technique, and you don't want to over extrapolate your data. Other questions?
Will this lecture be put on YouTube also?
Yes, barring any problems. The last time I tried to upload it, something had apparently gone wrong with eCampus, and I literally could not send you guys emails. I had uploaded it to YouTube. Has anybody looked at them?
I have.
Are they?
I have also.
Can you make sense out of them?
Yeah, they're good.
I know that sometimes getting the bar right relative to the slides doesn't always work out. I trust my voice is easy to hear. Are they beneficial?
Yeah.
Very much so.
Yeah. If I teach this again in the future, maybe I'll try to record my talks, even if it is live. I personally don't like teaching this way. I don't like staring at my computer screen without a give and take with you all, but that's sort of what we were forced to do. If I had to teach an online course, I'd probably also develop some kind of a chat room or something like that that would be independent of the lectures, so we could talk about things and maybe have it set up in such a way we could actually see each other, because so much information is lost when you're just hearing voices. I think oftentimes we don't appreciate how much visual information we use in our interactions with people. I sort of became very aware of it in a sense. First time I was in Scotland, and I found out that if I was not looking at somebody with a heavy Scottish accent, if I didn't see their lips moving, I couldn't understand what they were saying. They may as well have been speaking Russian. But if I could see their lips, I could understand what they were saying. And so it's just one example of visual cues, but there's lots of others. Body posture, how much movement's going on in a classroom. If there's a lot of movement, it usually tells you that people are bored, they're trying not to fall asleep. All of that's gone when it's just verbal. So I apologize. I hope everyone is practicing safe social distancing. This is just such new territory for us. Sweden has done virtually no lockdown, and their death rate is not that much different from, it's higher than other Scandinavian countries, but not that much different. They are aware of, and they're trying to avoid each other, large groups and things like that. But and then we have Hokkaido, which is an island, northern part of Japan, who was one of the first ones to go into lockdown. They saw the spread of the disease plummet, and so they said, okay, we can open things back up. They opened it back up, and sure enough, the rate of infection skyrocketed again. And so they had to go back into a second lockdown. And we're going to see each state is going to have its own rules and regulations regarding what can be open. I think the governor of Georgia is just asking for problems. I can understand opening up a restaurant and spacing patrons and disposable menus and things like that, ways that you can minimize any kind of cross-contamination. But letting massage parlors and tattoo parlors and, all those kind of things where by their very nature, there has to be very close, somewhat intimate contact between people. That doesn't make any sense. So we're going to see all kinds of different things. In this state, we've been fortunate so far. You can't see me, but I'm knocking on wood. Our incidence is fairly low. And the Governor Justice, whom I think overall has done a very good job. I think he was very proactive when he saw what was going on in other states. And so we went on a lockdown sooner than a lot of other states did. In fact, some states still haven't. I think he's handled this very well. I'm not necessarily a big fan of his on some of the things he's done in the past, but I applaud him for his efforts on the pandemic, dealing with the pandemic. But we're going to start to come out of it. And it's going to be slow, which I think it should always be. Restaurants are not going to be one of the first things. Movie theaters certainly are not going to be. Most places, although Georgia's an exception again, apparently movie theaters could open up, which is, potentially congregating 100 or 150 people, depending on the size of the movie theater. And depending upon the, thank goodness there's not a Star Wars coming out or something like that, where everyone wants, or Harry Potter, where everyone wants to go see it. Right now, movie production companies have essentially stopped sending movies out because there's no market. Because most of the movie theaters, not just here, but everywhere else, I guess with the exception of Sweden, we're very fortunate that we are at the stage we are in terms of science, particularly molecular biology, immunology, basically biology. Because if this had happened prior to our having a lot of the tools that we now have, it had just been a total disaster. When HIV was first discovered, they really thought it had to do with a homosexual lifestyle. I don't know why they didn't think of it as being an infectious disease. It took a while for them to realize that. I mean, there should have been a hint when it was, heterosexual Haitians and hemophiliacs that were getting blood products were also coming down with AIDS they should have realized that it was an infectious particle. It had nothing to do with the homosexual lifestyle. And when they found out it's a virus, there was a great fear that mosquitoes might act as a vector, which would mean that almost nobody was safe unless we went back to massive DTT spraying, which is DTT was something that was used during World War II and other times to kill mosquito populations. But it turned out to be very deadly for a lot of bird species, caused a thinning of the eggshells, which is why we almost lost several birds that are associated with waterways and whatnot. But if you have, at least so far, thank goodness, the coronavirus doesn't seem to be spread through any kind of animal vector. It's just in the air. We had one person, as I recall earlier, who said they were working on a wing of the hospital here that was dealing with patients. Are you on today?
Yeah, I am.
How's that going?
It's all right. We don't have as many people as I expected initially there to be.
Good.
They keep saying we're going to hit our peak, and that peak keeps moving back and back the more weeks we get in. So hopefully, it might not be happening.
Yeah, as you probably gathered, I'm something of a sports fan, even though there's nothing going on right now. But I still listen to Sportsline, which is on 1440. It's a lot easier to do it now since I'm home all the time. And about 5 minutes into the show, they always have an update of the situation regarding the coronavirus, both the infection rate as well as the number of deaths. And it's fluctuated around, but the number of positive tests compared to the number of negative tests is very low. It's less than 3% for the last 2 or 3 days, which is seen as one of the triggers to allow us to slowly start opening up the economy again. So do you have all the personal protection gear that you need?
Yeah, we still have, at least on my floor, we still have enough stuff. We're doing okay. They have limited how many you're allowed to have. I mean, I think we're down to, I think it's like 1 mask a week or something like that, but it's still not too bad.
How do you disinfect them?
The regular masks, you're not. That's like the regular mask, you're not. But the N95 masks, when you get transferred to another floor, they bring them back and do like a UV light clean on them or something along those lines.
Yeah, no, I've read about UV as well as temperature has been used to try and kill the virus. So, yeah, be very cautious. Don't pay much attention to social media and all these other scams out there about, potential cures and whatnot. Listen to scientists, physicians. Don't listen to quacks. If you're thinking about buying masks, you really need to be careful of the source. India apparently is turning back a lot of stuff from China because it's totally worthless. So anytime you have a crisis like this, there's always people trying to make money. There was a guy in New York City who had bought massive amounts of hand sanitizers and were selling them in like 1,000% profit or something like that. And so they're looking at prosecuting the guy. By the way, they say 70% ethanol is a good way to disinfect, which is true. If you didn't know, percent and proof are very different things. When you buy 80 proof liquor, which is what is sort of the standard in the United States, 80 proof bourbon, it's actually only 40% alcohol. The proofing actually came from when old moonshiners would mix gunpowder with a certain amount of the alcohol, and if it would catch fire, it was said to be proofed to be of a certain amount of alcohol. That's where the term comes from. I've heard rumors that certain individuals think as long as they just stay drunk, they're going to be safe. That's not true. You would have to drink so much alcohol that you'd kill yourself anyway. It's sort of like, Lysol, not a good treatment. Any other comments or questions?
How long until you grade exam 3 before the final so we can decide whether or not we should take the final or not?
Yeah, I mean, that's the problem with doing it like I did. Probably should have had this exam on Tuesday, in truth. This is why I'm going to send it out to you early on Wednesday. I'm going to have some instructions with it when I send it to you. Early meaning, you know, in the afternoon, because I've got meetings I have to go to Wednesday afternoon. But if you finish ahead of time, please send it on to me so that I can try to get as many graded as possible. I'm going to try to have them all to you by Sunday at the latest. If I don't, I don't know. I may have to think about postponing the final exam or giving different dates of the final exam for people who haven't gotten the feedback yet regarding whether or not they should take the final exam or not. Obviously this semester has been a mess for everybody, and I feel for you guys because it's not just my class, but it's all your classes. I'm going to do the best I can. I'm not going to put as much feedback on your exams as I did last time. I think one of the things that would help me, and I'm going to put it in the email when I send you the exam, is if you would put your name on your answer sheet, in the name itself. So it'll say MoGen or molecular genetics, exam 3, and then if I were doing it, I'd put Bishop or something like that. That would make life a lot easier for me to be able to sort and tell who I've gotten exams from, who I haven't gotten exams from, and also no PDFs or any other format. Just leave it in the Word document. It's much easier to grade. So there's a few things I hope to do to try and expedite the grading process. Probably the most important thing is not writing as much of a response to to why you lost points. I will be sending you the key, however, to the third exam, and you can compare your answer to the key. And if you want to challenge me or want clarification, feel free to send me an email and I'll respond to it. I won't, as I say, I may end up giving out half the final exams to, my feeling is in the past anyway, there hasn't been a large number of people that have opted to take the final exam, but the conditions were entirely different back then. So I may end up having to just give you individual dates for final exams. And I'm okay with doing that. I mean, I'm trusting you all not to be communicating with each other in terms of answers and whatnot. I prefer that system. I think in the long run, it's how you profit most is by your own work, your own accomplishments, not by finding out from someone else. I don't know if I told you all, but when I was in high school, state of Virginia, they had everyone, the whole state had the same chemistry exam. And it was an open book exam. And so we had a friend of ours who was pretty good in the clique I was in. And so he would open his book up and he'd write down the answers. They were all multiple choice. So he'd just write it down inside of his chemistry book. And then before we took our class, he had it first period. And I think we had it like third or fourth, 3 or 4 of us. We'd go and we'd get his book and we'd copy down the numbers he had. And people kept getting mad at me. They couldn't figure out why I was doing better than they were, because supposedly we were using the same answers. No, actually I wasn't. I was actually a better chemist, the guy we were getting the answers from. But I was too concerned with being labeled a nerd, I guess. So I tried to hide it by acting like I was cheating like everyone else, when in truth I didn't use his answers at all. So please don't do that. I enjoyed you all earlier. I hope everyone is healthy and safe. Again, I hate what has happened. As you all know, this is my last semester. I don't know how much longer I will have an office in the Life Sciences building. I don't know what is happening with the Life Sciences building. I haven't been back since you all left to the building. We had a lockdown shortly thereafter. They've done some disinfecting. Some people were allowed back in. People work with zebrafish, which requires daily maintenance. But the things I work with, you know, it's at this point it would just simply be frozen samples, so I didn't have to go in and tend to them or anything like that. But if you've seen my office, I've got months of work to do in my office to get it cleared out. If you're in the building, if they open up and fall semester, stop by. If you see my doors unlocked or partially open, stop by and say hello. Let me know how everything's going. Be safe. Have a good summer as best you can, but understand that even people your all's age are susceptible to the virus, and a lot of people have died your all's age. Not as likely as someone my age, but again, it happens. And even if you don't die, you can suffer greatly. As the gentleman who is here in Morgantown, I'm sure can attest to, who's working with some of the patients. Any other questions?
You talked previously about curving the class. Is there any more thought that you had on that?
No, I won't be able to curve it until I've gotten all of the exams graded, which is another problem with time constraints and the timing of everything, which again is why I'm going to try and streamline it as much as I can. Once I've gone through the exams, then I will know how you performed. If you perform like you did on the second exam, I probably will curve it somewhat. If you perform like you did on the first exam, there won't be any need to curve. So again, my target is low to mid 80s for the average and or mean on it. But unfortunately, I can't determine that until really I've got them all graded. So we feel free to communicate with me through emails and whatnot. I'm perfectly flexible to administering the final exam on a different date than Monday. Monday was what I'd originally targeted. The university changes the schedule in their infinite wisdom. Our final is actually supposed to be this Friday, but since it's essentially a take home, and I don't have to have the grades in for another week, we have, as far as I'm concerned, we have some flexibility in doing that. Any other questions? If you think of any questions, feel free to send me an email. I'm doing a pretty good job of monitoring my emails. Unfortunately, sometimes I miss emails. I foolishly used my work email address when I'd go to Best Buy and they say, give us an email address. So I'd give them my work address because that was the only email I was using. And because of that, I get a ton of just junk emails. And I keep trying to eliminate them. Some of them I can eliminate, some of them I can't for whatever reason. So be persistent with your emails. If you don't get a response within a day, send it to me again. Anything else? Again, have a good summer as much as you can. I hope all your exams go well, all your final exams. Stay safe, stay healthy, and hopefully you'll be back on campus in the fall. I'm not optimistic about having football in the fall, and I'm not even sure we're going to have basketball in the fall. It's really going to depend upon whether or not, the Sweden model is what we're going to adopt and just go ahead and accept a certain amount of infection, or whether we're going to see a bounce back like Hokkaido did in Japan, where you went from very low levels and suddenly you have an explosion of infections. And a lot of that's going to determine a lot of things, whether or not we have, in-person classes in the fall. Don't be surprised if they aren't delayed, if not going to online entirely. If we go to online entirely, it should be better for the fall semester because we will have longer to learn the technology and what we can do online. As it was, we were basically given a week to try and figure all this out. And I never used Zoom, YouTube, any of those kind of things before. But if you're given a whole summer, we'll improvise. We'll make it better. Anyway, take care. And hopefully I'll get to see some of you in the not too distant future. Okay, I'm gonna close it down if there's no other questions. Are there? No? Okay. Bye, folks.